John, for the kind introduction. All that talk about building even the defense make me say, where are the products that I can take so that my voice can be better? I've been struggling with my voice for the last 2 weeks, so I guess it's a segue to my presentation. So I'm here to present about the priority research program, and we call it in short, Elite Gut. So what is the consumer insight that support the concept of elite gut? We know the gastrointestinal tract health market is large and growing. This represents 60% of functional food. We know that GIT health is a rising concern for many consumers, in particular the worry well. Who are those consumers that worry well? This is people like you, and me, and you, that seek performance and productivity in the workplace as key priority. And this is important for Westerners, but more so for all Asian demographic. We know that Asian consumers have the highest stress worldwide. We know that stress impacts our GIT, and it's a major effect, as Richard pointed out in the video, on productivity and quality of life, particularly when it's experienced over a long period of time. Also, in terms of key consumer insight, we know that Asian consumers discuss more so about their problem with their GIT than Westerners. Also, they prefer to make decisions in terms of purchasing food and physiological health benefit. This key insight suggests and supports the concept of elite gut as an opportunity for food and health claim. Equally important, there are likely high adoption rate for this food. Okay, in summary, there are an opportunity that exists to market an elite gut that is highly desired by the Asian consumer to achieve their food potential. Improving performance and productivity of the very well consumer is the goal. So we know that the gastrointestinal tract is critical to the impact of food on physical, mental, and mental wellness. We know stress, as Richard pointed out in the video, to exam presentation, for example, cause variation of normal gastrointestinal function. This can include increased permeability of the gut mucosa, inflammation, change in transit time of how fast or how slow your food move in the gastrointestinal tract, to change in motility or secretion, and visceral hypersensitivity that lead to abdominal pain. There are more and more evidence that the microbiota have a key role, or the little minion present in your gut have a key role to play. As Richard pointed out in the video, the connection between the GIT and the brain is important. How you feel in your gut influence how you feel about yourself. Are you able to be productive to your day work? But the mechanisms are ill-defined. So what are the knowledge gaps? There are examples of food or ingredients targeting the gastrointestinal function and comfort that are approved by some regulators. James mentioned an example around DR10 this morning, around comfort, and there are as well in New Zealand around kiwi fruit, a cell-substituted alkaline. There are existing biomarkers of gastrointestinal function and comfort that are relevant to regulators. But there are insufficient knowledge of the food and health relationship around mechanism of action. we need to understand variation from the norm. As it happened, EFSA, regulator in Europe, accept irritable bowel syndrome as an appropriate grouping of people that can be used to support claim for the general population. So what is IBS? IBS is, as I said in the video, not a disease, it's a syndrome, and include abdominal discomfort and is recognized as a variation of normal function. There are different subtypes: constipation, diarrhea, and a mixed phenotype. But the phenotype is poorly characterized, and there are no robust clinical biomarkers. We need better prediction, and we need to understand the mechanism of action. So there are a few studies that have used more advanced omic technique to try to better characterize the phenotype or differentiate the subtype of IBS. In a study by Asen, they showed that 25 differentially expressed genes were able to distinguish IBS for healthy subject when looking at the colonic mucosa. Those genes was relevant to immune response and host gene defense response. In a more recent study, in using a larger cohort, 34 biological, mostly metabolite and gene, and 4 psychological markers were used. The biological markers were able to differentiate IBS from health. The combination of biological and psychological markers were able to increase that differentiation, and there was a small subset of markers that was driving this differentiation. And this suggests that it might be possible to have a panel of a limited number of markers that allow us to do so. In terms of being able to differentiate the IBS subtype, those biological markers were also effective. But the combination of biological and psychological markers did not improve markedly the differentiation. Other techniques that people have used, and we heard about them yesterday from Professor Gerd Cooper, it's looking at metabolite profiling. So there are few studies that have done that, and they're relatively small in number. So that have shown that higher level of some metabolite like amino acid, phenolic compound, and short-chain fatty acid that were present in greater amount in IBS fecal sample. But the results were not always in the same direction, and which often happen when you have small cohort. So in overall, few studies, small cohort lead often to conflicting results. So we need large cohort to increase the power to detect biomarker. And we need also to use combination of analytical stream to increase the coverage and the identification of those molecules that can be present in blood and other tissue or fluid. And in addition to that, we need a system biology approach that able to integrate those data and derive knowledge that we can use to identify mechanistic insight for IBS and how food can affect this phenotype. So briefly, this is a diagram that kind of summarizes what I just said. And I would not go in detail in the diagram, or John might push me off there. But basically, we eat food. Those food are breaking down and release metabolites, some or other compounds that are absorbed, some remain undigested, and we have those little minions or our microbiota that are very active and fit to do the job, and will turn those and ferment and generate metabolite. Some of those metabolites are good for us, others not so. And they can generate and potentially contribute to IBS. So the key point I want to make, if that works, is that in that review paper recently published, I think one of the key points was that we need more well-designed study that can study the effect of food intake on the generation of the IBS symptom. And I think a lot of the cohort studies that have been published, that's an aspect that have been actually not looked at, and that's something that we would take on board. So just briefly summarizing our hypothesis, as you heard up to now, the GIT is the critical component of the gut-brain axis. Communication between the gut and the brain underpin the beneficial effect of food on gastrointestinal function and comfort, with a flow-on effect to the brain. We hope by the research we do that to be able to eventually fast-track opportunity to develop food with validated GIT function and comfort benefit. So the research team, so in addition to myself, and my background is physiology, we have principal investigator Richard Geary from Otago University, who is an expert clinician in inflammatory bowel disease and IBS. And Richard will be speaking about some aligned research right after me. We have also lead researcher, and I think Liz has challenged me to find other adjectives to describe some of them, in biomarker identification and the use of omic technology. We have Carl Fraser, who also worked in Sally's program, who is a metabolomic expert, and Carl is an emerging researcher. And we have emerging researcher extraordinaire Wayne Young, that work in Microbiota, and Wayne have been trained with the best in New Zealand, Professor Gerald Tannock from Otago University, and one will look at the host and microbe interaction, and as well use his extraordinary skill, as Liz pointed out, to integrate the data. And we have my good friend Elizabeth Bloom, who will provide expertise around the immune response, and this is from the Malagan Institute, and also Janine Coney from Plant and Food, who will be speaking later, and very keen to tell you about plasma neurotransmitter analysis, and that will be quite good for us in terms of linking the GIT tract to the brain. We are also establishing international collaboration, one with Professor Paul O'Toole from the APC Microbiome Institute in Ireland, and it's around the microbiota, and also with Richard, I have a collaboration with Nick Talley in Australia, and Nick is, thousands of publications, and with a lot of expertise in IBS cohort and relevant biomarker. So in objective 1, which is led by Richard, the aim is to increase the predictability of functional IBS phenotype. We will recruit subjects into a cohort that we conveniently name comfort. So we will have a group of subjects that will be healthy, but they will have gone for colonoscopy, for screening and surveillance, but won't have GIT symptoms. The grouping of IBS subjects will have functional symptoms and require colonoscopy. We'll get a range of data, some qualitative, some biological data, like demography, diet, quality of life, psychology, and also sample for biomarker analysis. What we hope to achieve is that to confirm that existing biomarker and the qualitative information will allow us to further characterize, using a system approach, the IBS phenotype. If we can achieve that, and we will be able to better design a nutritional intervention to establish a food effect on relevant biomarker. In Objective 2, and that's where most of the work done by myself, Wayne, Carl, Liz, and Janine fit into it, we will identify microbial and host factor to provide mechanistic insight. This includes gene expression in blood human cell, metabolite profiling in a range of tissue or fluid, microbiota profiling, and also the integration of the data. So the next few slides, I will just go through a few techniques that we'll be using. The first is around metabolite profiling. For most of you, you probably are aware, but in case some people are less familiar with that type of work, there are essentially two types of methods, non-targeted, or often referred as metabolomic, or targeted. Carl Fraser, who is here, but not here because he's on sabbatical in France, and doing more work on biomarker that will be very useful for Elite Gut, Sally's program, and other program in the Contestable project. So non-targeted or metabolomic is a semi-quantitative detection of a wide range of metabolite without a priori knowledge of what you want to look at. It has minimal sample preparation and to ensure no losses of metabolite. And in contrast, targeted is targeted, absolute quantification, and as specificity. On this picture, we have Janine Coney, who will be speaking to you shortly, and who use a targeted approach. So just briefly trying to, I guess, drive the concept why it's important to use multiple approach. We have to use targeted approach, but also untargeted approach. On the first panel, that way, you have a pathway that have 6 metabolites. If it's working normally, each of those metabolites have a certain abundance. If the pathway is blocked, which is that one there, then completely blocked, well, you will have accumulation of metabolite above the blockage and no metabolite in the below. But life is not that simple. Most of the time, you will have a pathway that is slightly modified, and the body have ways to that an alternative pathway will be used, and so you will have a different outcome in terms of pathway. Also, sometimes it's really messed up, and it's a range of change that happen that we don't really understand. So if you do targeted analysis, you will measure those 6 metabolites. If you do untargeted, you will be able to measure the 6 metabolites, what's happening to them, how much, what is their abundance. In addition, all the other metabolites that might be responsible for what you're observing in terms of phenotype. In terms of the microbiota, there's several techniques that can be used. We will be focusing on understanding what are the genes that are present and their functional potential. This technique is called metagenomic. And also we will be focusing on metabolomic to look at the end product of the metabolic interaction between the host and the microbiota. We will store some sample to do other type of analysis, like looking at bacterial gene expression, but we don't have enough money at the present to do all of them, so we pick the key one that we think that will help us to build the story. So as I mentioned earlier, all those techniques are data intensive. They generate huge amount of and require intensive computer. But we have workflow for that, and that's not the roadblock. The roadblock is really the ability to be able to integrate those data to derive knowledge, to know that the genes that are differentially expressed, the metabolites that are increased in abundance or decreased in abundance, the change in the microbiota composition and function, all together are responsible for the phenotype of IBS or other conditions we observe. So we hope by doing that we will be able to increase the predictability of existing biomarkers, to have a panel of biomarkers that might have been derived with a lot of data, but a limited number that we can use that give us, in terms of characterizing the phenotype, specificity and sensitivity. So we know that when we look at those markers, we can predict what will happen. So better characterization, more sensitivity, quicker and cost-effective. In Objective 3, we want to take all the learning from Objective 1 and 2 and do a proof of principle of food intervention. So using the cohort and probably a subtype of the IBS, we will have to see, and probably the selection of the food will come with those that have strong preclinical evidence or even early clinical evidence. And where we will get those data, obviously there are some published evidence, but they are aligned research across the range of organizations on New Zealand that we will be able to discuss with people that do that research. We have an industry reference group that able to tell us, well, this is that food option, yes, all that evidence, but does it actually make sense in terms of a food that will go on market eventually? And so using our core, using the biomarker that we have developed and validated, we aim to demonstrate increased predictability in that relationship between food and GIT health. Timeline, always important. I won't go through the whole slide because John is looking at the clock. The main point, I guess, I want to indicate here is that the line here indicates in gray the regular meeting that we have with our industry reference group, and with the red block indicate where we will have, and it's an estimation when this will happen, obviously, we'll have key decision point influencing what the research might be doing in the following year. But the key work now, and I'm pleased to say that Richard sent me an email yesterday with a letter saying we have ITSIC approval to proceed, so we are on the go. It's the recruitment of that cohort, which will be a unique opportunity for New Zealand to use in the later year, and also the stratification of that cohort. Also, the other major piece of work is the work around the characterization using omic technology, and obviously the food principle that is toward the program. So we have been tasked by the government to the HVM host in Auckland University to develop capability, bring together people that have not worked together before. And we have done that. I think this was the long run for the 2.5 years to get there. The programs are on the way. But the ultimate goal is to work the industry from the get-go, and develop the research to assist them to design, develop, and validate food to support an elite gut for the very well Asian consumer. 1 minute to do the acknowledgement. That's good. Okay, I will let loose, John. So before finishing, I want to acknowledge a range of people. First and foremost, the HVM management team. David, John, Ephraim, Eli, Eleanor, Polly, and probably others that I cannot remember. I think those guys are the unsung hero of HVM. And it's been a great pleasure for me to work with my colleague from the various priority investment, but with those guys in management. Even so that sometimes the email was a bit demanding, but that's okay. Also, a great pleasure to work with the Gut and Immune expert panel. This was a really productive journey. At the beginning, the Gut and the Immune program were together. We've been separated together, and eventually, I think we have 2 strong programs. So I think that's a credit to that expert panel that really had discussion that were really fruitful and challenging, but fruitful. Also want to acknowledge other priority program in HVM, consumer insight and science of food, which are critical in us as well, in addition to the industry reference group, to see what is possible in terms of food solution, how we preserve a bioactive that might be relevant in a food product. Thank you very much.